Hello students, in continuation to our last class, we need to study the graft rejection. When any person infected severely in an accident or infected severely, then one or few of the organs may be damaged and they may not work properly. So in such situation, such organs must be replaced from a suitable donor. So such type of reactions we are calling as a organ transplantation. Normally, human organs like liver, kidney, heart, skin, cornea, they may be using for the organ transplantation. And even that organs we are calling them as a grafts also. So a graft is also called as a tissue and organ which can be transferred from a suitable donor to a recipient or a patient. Now, the suitable donors may be an animal, or it may be a primate, or it may be a human being. After successful organ transplantation, the person may be sometimes are going to be undergone a graft rejection. What is a graft rejection means? The immune system may identify such type of graft, okay, and they will be identified as a non-self and they are eliminated from the body. So such a type of reaction we are calling as a graft reaction, which is under the influence of T cells actually. So in the graft rejection, the graft is going to be eliminated from the body, and even the subsequent reactions are very dangerous to person, and that leads to the person's death also. So to avoid all such conditions, the graft rejection may be avoided with the help of some few techniques. Suppose if you observe, the blood group matching between the donor and the recipient is important, and tissue matching also. In the tissue matching, on the tissue surface there is a major histocompatibility complexes. If they are matching, so then it won't be identified as a non-self. So these are the two important things before to the organ transplantation reaction they are going to be observing. So both are essential. Next, when you observe the graft organ transplantation, okay, so in that condition, the pre and post operative steroid therapy is important. The person who is undergoing the surgery may involve pre and post operative steroid therapy. A steroid is a group of chemicals, okay, so which directly react with the immune responses and they may suppress such reactions if there is no reaction and there is no rejection also. So such type of treatment we are going to provide to such patients actually. And even one of the treatment we are giving the immunosuppressive agents, example cyclosporine. So they may be given after the operation or before to the operation to such persons. Next, there is a different kind of organ transplantations we are going to study. So in that xenograft, suppose an organ transplantation is in between the different individuals of different species, then we are calling it a xenograft. Autograft indicates the tissue or graft which is transferred from one place to the another place of the same organism. You may be observed in case of burn, fire accidents, so the skin which was affected area, okay, so that may be replaced from the and it will be taken out from the normal skin and it will be covered on the affected area, so it is an autograft. Then allograft is different, where in between the individuals, okay, who are related to the same species but the genetic makeup will be different, that is an allograft. Allo indicates a difference at the species level, different at the genetic level, but the species is same, so it is an allograft. Then isograft indicates the graft between the individuals who are genetically identical, like the twins, genetically identical twins. Okay, so they may be related to the same species also. That is isograft. Now I am showing the graft rejection phenomena with the help of a pictorial diagram. So he is an affected person. Suppose if we take example as a his kidney was affected, then the kidney transplantation must be takes place. So how it will be? So we are taking a suitable donor. A suitable donor definitely is a person, or he belongs to the human beings only, because most of the cases like donor recipient, there is a match between the blood group and even tissue matching also. If both are matched, then only there is a successful transplantation reaction. So the donor will donate the kidney and the recipient will receive. And afterwards what happened? Let us observe carefully. The recipient, if the recipient T cell interaction is very high, then the graft will be rejected and even it may lead to death of that person. To avoid this situation only, the same recipient was treated with the steroids, which we are calling pre and post operative steroid therapy. So if we inject with the immunosuppressive agents, then the T cell reaction will be reduced and there is no graft rejection and the person will lead a normal life. So these are the relation to the graft rejection.
We studied that the acquired immunity is a specific type of immunity which identifies the pathogen specifically and eliminates from the body. The acquired immunity is divided into two different types, that is an active immunity and a passive immunity. They are again subdivided into natural active immunity and artificial active immunity. In the same way, the passive immunity is also divided into natural passive immunity and artificial passive immunity. Now, let us understand these topics in detail. What is an active immunity? When a person's own cells produce antibodies in response to the natural infection or vaccination, we are calling that immunity is an active immunity. So what happened originally? The person was naturally infected. Means there is an entry of a pathogen which containing antigens. In response to that antigen, immune system has produced the antibodies. There is an antigen antibody interaction and they may be eliminated from the body. So such kind of immunity is a active immunity. And what is a vaccination again? A vaccine introduced into the body which elicits the production of antibodies. Such kind of immunity is called as an active immunity again. The active immunity properties, if we observe, it is a slow process. Why? Because the antigen interaction and the antibody production and the interaction between the antigen antibody elimination, so all this process will take a lot of time and even it is a slow process again. If you observe, it takes lot of time to give immune response, that we already observed, and it is a long lasting, means the person is protected for a long time from the infection and even from the subsequent infections also. That is a long lasting, and the active immunity is a harmless to the body. Okay, so these are the properties. The active immunity is divided into two different types, that is a natural active immunity and artificial active immunity. Let us observe what is a natural active immunity first. When the person recovered from diseases like smallpox, measles and mumps, the same person won't get the same infection on subsequently. Why? Because the person already got the immunity against that infections. Even in our day-to-day cases also, we may observe the person once got the smallpox, he won't get the same infection in his lifetime. So that is the smallpox, measles and mumps case. Okay, and such type of active immunity we are calling natural active immunity. If you go detail into that topic, the person's immune system is developing the memory B and D cells against the smallpox, measles and mumps, and even in the subsequent infection also they will fight against such pathogens and he won't be getting the disease again. That is a natural active immunity. Coming to the artificial active immunity. Resistance induced by injection of vaccines. Okay, so what is a vaccines? First we have to know. If you observe the definition, the vaccine is a preparation of inactivated or attenuated or weakened pathogens, or sometimes it may be antigenic proteins of the pathogen. If you observe the pathogen surface structure, there is a number of proteins present outside. They may induce the antibody production inside the immune system. So this all we have studied previously in the acquired immunity. Okay, so first what we have to know, the pathogen must be inactivated. That means the pathogen may be taken and will be killed. That is nothing but killing or inactivating. Next, attenuated or weakened pathogens. The pathogen may be weakened, means it won't be causing any disease, but it can induce the immune response. That is the importance. Okay, so attenuation indicates it is weakened. It can induce the immune response, but it can't cause the disease. Okay, so such a preparation we are using as a vaccines. And I am giving a number of examples which are very important for the NEET examination. Carefully observe. The live form of vaccine means the attenuated form of pathogens we are taking and injecting into the person. Okay, so examples are BCG vaccine for tuberculosis. What is the full form? BCG, Bacillus calmette-guerin. Okay, so it is against the tuberculosis. Sabin vaccine, which is a polio vaccine actually. So the attenuated form of poliomyelitis we are taking. Okay, and it will provide protection against poliomyelitis. MMR vaccine, full form is measles, mumps, rubella. So within a single vaccine it is going to cover the 3 diseases at a time. That is the MMR vaccine. So if you observe all these vaccines, they are nothing but live attenuated form of pathogens which provide immunity. So such kind of immunity we are calling artificial active immunity. Okay, now the killed vaccine means the pathogen is taken and it is killed and is used as a vaccine. Let us observe TAB vaccine for the enteric fever. Typhoid, paratyphoid A, B, that is a full form. Okay, and enteric fever is a type of fever caused by Salmonella typhi. Sac vaccine for polio. We know the polio is a viral disease. Sac vaccine, in the honor of Sac we have given the Sac vaccine actually. And if you observe the toxoids again, toxoid is a type of preparation which contain inactive form of toxins. Normally diphtheria is a disease caused by Corynebacterium diphtheriae. When it is entered into the body, it produces number of toxins. So that toxins will cause us diseases in the body. To avoid such case, we are taking out that diphtherial toxins and inactivating them, and these inactivated toxins we are calling as a toxoids. Suppose if you introduced the toxoids, they will be entering into the body and they produce the antibodies and they provide immunity against the diphtheria bacteria. In the same way, tetanus also. Tetanus disease caused by Clostridium tetani. Okay, so the tetanus toxins we are taking, and they are preparing as a toxoids in the lab, and later they will be injected into the patient. So all these are type of vaccines. Last and but not least, it is very important, that is a subunit vaccine or recombinant vaccine. Hepatitis B vaccine can be produced in the laboratory conditions, and it is a recombinant vaccine. We will go detail into the production of hepatitis B vaccine later, but now the vaccine we are going to study against the hepatitis B. Now DNA vaccines also present, where the pathogenic DNA we are taking, inserting into the body, and that will produce the proteins which will provide the resistance. That is about the DNA vaccines. Now for easy understanding, I am giving you a pictorial diagram where here the person was injected with any kind of vaccine. Take any type of vaccine which is injecting here. Okay, so what is a vaccine originally? It is made up of antigens. Now the person is exposed to specific antigens. The immune system will produce the antibodies which are specific to that antigens. Now what happened? There is a formation of a primary immune response because the person is exposed to antigen for the first time in his life. So there is a primary immune response in that body. There is a fight against the antigen and the antibody, and they will be eliminated. Meanwhile, there is a production of memory B and T cells also. Now what happened? The person won't get disease because of the vaccine, but the immune response is protecting him. So what is the immune response now? The immune response containing memory B and T cells against to these antigens. That is nothing but the secondary immune response is going to be start when there is an entry of pathogen. So what happened? When the pathogen is entered, they are activated, they produce the antibodies, there is a fight against between them, so the person is protected from the disease. This is the mechanism behind the vaccination. Now we are calling such kind of immunity as a artificial active immunity. Hope you understood properly now. So the resistance induced by the vaccines, when they are injected into a person, we are calling it is a artificial active immunity. Now the active immunity part got over. The second part is nothing but the passive immunity. When we are injecting a person with the ready-made antibodies, then that person will be protected from a foreign agents. Okay, now if you observe carefully, the immune system is not directly participating in the reaction, but the preformed antibodies or ready-made antibodies. Means antibodies are produced somewhere, but they are now transferred to the patient. So what happened? There is a direct fight between the antigen antibody interaction, and there is no involvement of the immune system here. That's why we are calling passive immunity. Okay, now if you observe carefully here, there is a properties I have given. It is a temporary type of reaction. Why? Because there is an immediate relief, and even it is a not long lasting. Okay, so temporary indicates the immune system is not directly participating, and even there is a production of memory B and T cells. Okay, so but the relax will be up to some certain time only. That's why it is a temporary relief, and it is a not long lasting. The person won't be protected for longer time. Okay, that will be not long lasting, and it creates problems certain cases, but not in all. In certain cases it may create some problems. This about the passive immunity. The passive immunity is of two different types, that is a natural passive immunity and artificial passive immunity. In natural passive immunity, the resistance is transferred from person to person by means of antibody transfer. One example, during pregnancy, from mother to fetus, there are transfer of IgG antibodies through placenta. So what happened in the case of mother, the immune system is transferring the IgG antibodies to the fetus. So the fetus is protected from further infections. Second example is after the childbirth, during initial days of lactation, the mother secretes colostrum. Colostrum is nothing but yellow fluid which is rich with the IgA antibodies. When they are going to secrete means in the initial lactation period, means milk secreting period we are calling as a lactation. So when she breastfed the child, then the colostrum antibodies are transferred, so the infant will be protected. If you consider, this passive immunity is a temporary but not permanent. Why? Because the antibodies are existing for few months only. Okay, so next we have to study about the artificial passive immunity. When the persons are in the critical illness, like when they are infected with the tetanus, diphtheria diseases, they require an immediate clinical treatment, and even in the snake bite case also. So such persons are administering with the preformed antibodies or antitoxins or by means of anti-venom. Okay, so in all these cases, the antibodies are transferred from sensitized person to the normal person. So the person will get the immunity. Okay, so such kind of immunity we are calling artificial passive immunity. Now let us observe what is the antitoxins. Antibodies raised against to toxins we are calling antitoxins, and the anti-venom or anti-venin is against to the venom of the snake bite. Okay, so how it is produced, the antitoxins, that I have shown with the schematic representation, carefully observe. It is not that much important, but we have to understand how the antitoxins are produced. Okay, so the horse normally may get the tetanus. To prevent the tetanus infections, they are injected with the tetanus toxide. Is nothing but vaccine only. So vaccine again, it is an antigen only. So because of the horse immunization, the antibodies are produced. Okay, so antigen recognition and the immune system produces the antibodies. So such a mechanism we are calling active immunization. Okay, so we have to take out the blood from that horse and we have to purify the serum with the antibodies only. So such kind of antibodies we are calling anti-tetanus serum. Okay, so we have to transfer that anti-tetanus serum injections to the persons who are infected already with the tetanus, so the person can be recovered easily. So this procedure of transfer of antibodies from sensitized organism to the person infected is called as an passive immunization. So we are observing the differences now. Active immunization is nothing but deliberate inoculation of microorganism or antigen to produce the antibodies is called active immunization. But the transfer of antibodies from organism to organism is called passive immunization. Now let us observe what is a vaccination. A vaccination is nothing but a vaccine given to person to protect him from the further infections with the same pathogen. So here the vaccine is nothing but the antigen. We already discussed the pathogen. Okay, weakened pathogen or attenuated pathogen we are going to inoculate, and there is a production of antibodies, memory B and T cells, and the person is protected subsequently. Okay, so that is a vaccination mechanism. Then passive immunization. We already observed that person injected with preformed antibodies is called passive immunization. Now there is a similarity between the vaccination and active immunization. So they are based on the memory of immune system. Both the memory B and T cells are producing and they can fight against with the subsequent infections. Okay, now the next part we have to study, that is recombinant vaccines. One example in the textbook is given, the hepatitis B vaccine production, where the bacteria and the yeast cells are utilized for the production of hepatitis B vaccine. Okay, so how the recombinant vaccines are developed, that we have to observe. There are two techniques called genetic engineering and the rDNA technology we are using to produce the hepatitis B vaccine. These two are going to be studied in the biotechnology principles processes chapter, but for easy understanding I have given the figures also. Right, carefully observe here. The hepatitis B virus I have given, the virus coat, protein and the DNA. Okay, and you know the DNA is containing number of genes. Okay, one of the important gene which produces the antibodies that I have selected, that is the hepatitis B antigen gene. Now from the bacteria Escherichia coli I have taken the DNA again. Both these DNAs are clubbed together, combined together, forming the hybrid DNA. This hybrid DNA may be transferred to the bacteria or may be transferred to the yeast cells. Why? Because the copies should be multiplied. Okay, so the yeast cell is now undergoing the binary fission. When the cells are producing or multiplying, the DNA also multiply, and at the same time the DNA produces the protein also. So we require only the hepatitis proteins. Okay, so we have to kill that yeast cells and take out that proteins only. We are transferring into the vials and then we are using for the injection purpose also. So this is the production of hepatitis B vaccine.